Good afternoon, everyone. Thank you for coming to our talk. My name is Sylvia Doiguray, and this is my lab partner, Amelia Smith. And we are very excited to talk to you about our experiments today concerning cell migration under microgravity conditions. Okay, so cell migration is an extremely essential process from birth to death and every process in between. So in the human embryo, this ball of cells have to be able to migrate in order to allow for the embryo to develop into an actual organism. In addition, white blood cells that fight infection, they also have to be able to migrate in order to actually capture bacteria and other foreign bodies and engulf them. Also, cell migration is important in wound healing and immune response. So when cells have, during wound healing, cells have to actually be able to invade the wound for the wound to actually heal. So that occurs via cell migration. Okay, so I want to give you a brief overview of the actual process of cell migration. So what actually happens in cell migration is that a cell reaches out at one end, adheres or sticks to the surface it's on, which is usually other cells, and at the other end it contracts, and that allows it to push forward. And so the proteins that are important in this process are actin and E-cadherin. So actin is a protein that is in the cytoskeleton of a cell, and so actin, when it grows, this allows for the cell to actually reach forward, and when it contracts at the tail end of the cell, it actually allows it to move forward. And E-cadherin is the protein that allows the cell to adhere to other cells and stick and stabilize itself while it migrates. For our experiment, we focused on both of these proteins, actin and E-cadherin, and an activating protein that begins the process of cell migration. And we wanted to look at all three of these components and see how each of them individually and together affect the cell's ability to migrate. For our experiment, we used fruit flies, so that was our model organism, and we used fruit flies for a few reasons. The first is that fruit flies have a very short generation span, so they're easy to work with because of that, so they have a short life cycle. And in addition to that, a lot of the genes in fruit flies are similar to genes in humans. And finally, in the fruit fly ovary, there are migratory cells that we actually were able to look at and look at these proteins, actin, E-cadherin, and our activating cytokine, and study how they're affected.
So since we're interested in how microgravity affects human cell migration, within our system, we're looking at how microgravity affects the border cell migration in the fruit flies. And so our questions here are, does microgravity delay cell migration? And if it does delay cell migration, what component of the migration is affected? Is it the actin, the ECAD, or the activating protein? So our team chose the FME lab that had 2 clamps, creating 3 separate sections. In the first section, we would have fly food and 5 female fruit flies with white eyes. In the second section, we would have fly food and 5 male fruit flies with red eyes. And in the third section, we would have a fixative. It's 4% paraformaldehyde. And upon arrival at the International Space Station, the experiment will begin when the first clamp is removed, allowing the female and male flies to mate and give rise to offspring that will have only developed in microgravity. Then, towards the end of the mission, the experiment will be concluded when the third clamp is removed, allowing the paraformaldehyde to fix both parents and offspring. Upon return to us here on Earth, we will differentiate between parents and offspring based on eye color, and the offspring will be dissected and stained to see the migratory cells, the border cells, as well as actin and E-cadherin. We will score for delayed migration, and if we find delayed migration, we will be able to compare the actin and E-cadherin networks to the ground control. And we hypothesize that we will find delayed migration, and then on top of that, we expect to find disorganized actin and altered levels of activating protein and E-cadherin. If we find delayed migration, we hope that this will motivate researchers to look further into astronaut immunity because cell migration is very crucial in that process of fighting infection. This concludes our presentation. We'd like to thank the NCE-SSE, the STARS Guyana Laboratory, and the University of Maryland College Park and Dr. Daniel Serrano for the opportunity to participate in this program. We'd also like to thank the Fly community for stocks to experiment with and the Bloomington Stock Center, as well as the other organizations listed there for funding.